From MicrobeTV, this is TWiN, This Week in Neuroscience, episode number 65, recorded on October 20, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New Orleans, Vivian Morrison.
Hey, guys.
How are you today?
I'm doing very well, thank you.
Is the weather wonderful down there?
It is so nice. It's cold. It's, 78 degrees. It's cold.
78 degrees.
Actually, I have no idea what temperature it is, but it's really nice.
Here it's 16 C.
Okay.
Which I think is 60-ish. Also joining us from New York, Tim Chung.
Hello. Hi, everyone. Yes, it's a quite nice day today, actually. Temperature is pretty good.
This weekend was very warm.
Yeah. It's. I thought we're supposed to be heading towards winter, but I'm not complaining.
Winter?
New York doesn't have a real fall. You've seen those Instagram memes when fall is a rapidly developing. It takes many, many steps. And then there's one day of fall, and then you go right into winter. You have fake fall. You have fakes. It's very funny. Anyway, if you like these programs, we'd love your support. We don't do advertising. We just depend on your donations to enable us to make these programs. And MicrobeTV is a nonprofit. So in the U.S., your donations would be federal, U.S. tax deductible. Although, for me, it doesn't matter. You can have all the deductions you want. It's never more than the standard deduction. But maybe you're better than I am. But still, we'd love you to donate just to support the dissemination of good science information. That's what we do on these programs. Go to microbe.tv/contribute. Now, it's very interesting that a theme on TWiV has been dementia and viruses. Mm-hmm. And Friday, we did a paper showing that reactivation of varicella-zoster virus is a risk factor for dementias of all sorts.
That being shingles and chickenpox, right?
Right.
Yeah. And
so does Shingrix help?
Well, Shingrix and the other one, whatever the attenuated vaccine, which is older, that helps. So it really makes a difference. It reduces the incidence of dementia remarkably. And it was a really big study. And they controlled for all the confounding factors. And that's a problem with an observational study, which we'll see today. You can never eliminate all the confounding factors. So an observational study, you can never conclude that X causes Y. Yeah. You can never conclude that Tylenol causes autism. Never. Even if you had great data. All you could say is that there's an association. That's and right. Then you'd have to figure out another one.
If there were one.
If there were one. But of course, in the case of autism and Tylenol, there is none. But so that is a good question. If you find no association, do confounding factors not play a role?
Could they be driving the fact that there's no?
Could they?
Yeah, I bet you. Same way.
Confounding factor could obscure some true underlying effect. Yeah.
So it can work either way if you're looking for an association or not. Okay. Mm-hmm. But anyway, you can get around that. In this paper, they had multiple hypotheses that they tested, and they're all internally consistent. Or you could also do experiments in the lab that could, support.
Or if you're the president, you can get your population to try to do an experiment by telling them to stop taking Tylenol when pregnant.
Yes.
But I mean, if he had a bit more if he was a bit more organized and did it seriously, he would actually get people to keep track of when they stop taking Tylenol so that we can collect some data.
Yes.
But he just laid it out there and said, stop taking Tylenol.
So I had 2 doses of Shingrix. So after the TWiV, I was very happy. But now today, you're going to see that there's more than one cause of dementia.
We're all screwed.
We're all screwed. Yes.
Sorry, listeners. That's it. Takeaway message is we're all screwed.
I thought the same thing when I read this paper. We're all screwed because maybe we should be hunter-gatherers but we'd still be screwed because the lions would eat us, right?
Yeah. You die of exposure, because in the paper they have a map where they show the air quality, because it's what we're going to be talking about today. And it's really interesting. Makes sense. There's some parts of the country where it's like the air is pristine, other parts not so much. And then see how that overlaps with the condition that they're looking at is pretty interesting. And actually, it wasn't, if you looked at those maps, it didn't look like it was obvious.
Anyway, the paper is going to be presented by Tim. Another paper about dementia.
Okay, should I start? let's get
that was your lead-in.
Okay, so today, I think we got a bit of setup already. So today we're going to talk about this paper that came out in the journal Science quite recently in 2025 September, and it is called. The title is "Lewy Body Dementia Promotion by Air Pollutants." And let's go to the authors. So there are, I think, 4 first author, and then a million other authors. They're like 1,000 authors here. The first authors are Zhang, Liu, Wu, and Jia. Sorry about the pronunciation. I speak Cantonese and not really Mandarin. That's blame my parents. Don't blame my parents. Blame me. And 3 last author, I believe, and that is Ted Dawson, Shi Zhonghan, and Xiaobo Mao. And these authors are, by and large, I think, from Johns Hopkins in the US and
also Columbia.
And also Columbia. Yeah, I think that covers.
And there's one from New Orleans.
There is? Oh, no.
No, there is. And then I was worried that in the paper they were going to say something about how New Orleans is like the worst place to be ever because of, Cancer Alley.
It does look, is that what it is? Oh, no.
Yeah, there's a lot of chemical refineries and stuff.
Okay, so there are actually, I see a lot of other places like Georgia Tech, Texas Health Science, even Imperial College and University of Cambridge, shout out UK. So there's very much an international group of people, actually. As you can tell, they're going to gather data from around the world. But we're going to talk about, 2 things that we're going to talk about, which is 1, Lewy body dementia. So I'll explain what that is. And the second one is air pollutants, which probably is self-explanatory. But the reason why I picked up on this paper, so a slight, another intro is, just for listeners' benefits, I actually do research on Parkinson's disease. So that's my day job. And if you work on Parkinson's disease, the one celebrity actor that is a champion on Parkinson's disease that everyone knows about is Michael J. Fox, aka Marty McFly, who got Parkinson's very, very young. I think he was like 29, below 30 when he was diagnosed, which is very atypical. And then since then, he has a Michael J. Fox Foundation, which is a foundation that both raises awareness for Parkinson's disease and also does funding and also do research grants for Parkinson's disease. So Michael J. Fox has been a huge champion to promote awareness about Parkinson's disease and also help research. But this maybe, at least for me, the second person I know that comes to mind when we talk about Parkinson's disease, bit of a stretch, is actually Robin Williams, which I don't know if listeners, if they're a bit younger, they might not be as well known to them. But to me, Robin Williams is Genie from Aladdin, and he's also Peter Pan from Hook.
Such a good movie.
But other listeners might know him as I don't know what his name is in the film, but he's the psychologist in Good Will Hunting. He plays a lot of
he's so many things,
a lot of teacher character. He's like Patch Adam, the doctor in Patch Adam.
Didn't he play a woman in one movie?
It's Mrs. Doubtfire.
Doubtfire, right.
He plays a cross-dressing dad. But the movie that actually I have been aware of but never watched until I decided to do this paper is this paper called Awakenings, which is based on a book by the neurologist Oliver Sacks, which maybe some of the listeners have heard of. So in Awakenings, Robin Williams plays a neurologist who came across, in the 60s, who came across all these patients, one of whom is played by Robert De Niro. And all these patients suffered from this disease called encephalitis lethargicus. So these patients suffer from brain inflammation in the past that make them lethargic, make them not move. And Robin Williams in this movie, in this film, kind of shows up to a neurology ward, and all these patients don't move, and they haven't been moving for 20 years. And then Robin Williams go around trying to, being a new doctor, look for ways to help them, and then found this drug, recently discovered drug called levodopa, which is a precursor to dopamine. It gets converted to dopamine in your brain. And he gave all the patients L-Dopa, and all these patients went from kind of statuette, not moving, to like becoming alive again. And they were, dancing around on the dance floor, playing baseball, all kinds of stuff. And it was a wonderful film. It is a little bit sad because it turned out the effect of levodopa was short lasting. It worked for one summer and then for still unknown reason went away. But it's a very interesting movie to watch. I recommend.
But that's a real story.
It's a real story. Like, encephalitis lethargicus is real, and people we still don't know what causes it, which is scary because it can come back, it can reappear again. But according to Oliver Sacks, he thinks there's some evidence, which is still debated, that it's an enterovirus. So Vincent, you might be interested, but there's no way of knowing because we don't. I mean, maybe we can go to look at some autopsy samples and try to look for viral particle, but I think it's going to be hard.
It could be a hit and run too, and the virus is long gone.
Exactly. But it's very. It suggests that Parkinson's symptoms can be caused by many things. But anyway, that was quite a digression. But I brought it up because Robin Williams, who plays a neurologist who diagnoses these patients with kind of Parkinsonian symptoms, he himself actually, it's a very sad story. He took his own life, I think maybe around 10 years ago. And before he died, he suffered from like severe depression. And also, apparently, it very rapidly lost his memory and suffered from a lot of anxiety attack and deep fear. And also weird symptoms like insomnia and like maybe slight tremor. And he was also acting. He was working at that time, and he went from being able to do like Broadway shows, like seasons of Broadway shows with like remembering like an hour worth of lines, to like forgetting single lines in a movie shoot. That made him completely like, if you can't even do that when 100 people depend on you for a job, that's like, you can imagine how much of an emotional toll it took on him. So he suffered from severe depression and took his own life. And there's a letter submitted by his widow, Susan Schneider Williams, to the journal Neurology that details kind of the episode of his death. After Robin Williams died, the pathologist did an autopsy on his brain, and they found in almost every neuron that or maybe even every cell of the brain that they examined, I'm guessing it's neuron, every neuron that they examined in the brain, there was this pathology called Lewy body. There's this clump of protein that stains for, the clumps of both protein and lipid, that it stains positive for a particular protein called alpha-synuclein. And they clump up into these thing that neurologists have called Lewy body inside neurons. And it is completely widespread in his brain. Apparently, it's one of the most severe cases of Lewy body disease the neurologists have ever seen. And it was like a semi-miracle that Robin Williams kind of held it together for that time. And that explains pretty much all his symptoms before he died. So,
how old was he?
Sorry?
How old was he?
I can't remember.
I'll look it up.
Yeah.
I'm just curious how long he survived.
I'm guessing maybe 60s, in his 60s perhaps.
Yeah, that's what I was thinking.
Yeah, 63. Yeah.
Yeah, it's quite young. So this is really... so it was really hard for doctors to diagnose Lewy body diseases, Lewy body dementia, because you usually diagnose it after autopsy to confirm with staining, labeling against this thing called alpha-synuclein. So Lewy body dementia shares something with Parkinson's disease, actually. That's which is another link to the movie Awakenings. In both Parkinson's disease and in Lewy body dementia, there's a telltale sign in the brain, usually established after death in autopsy, which is a proliferation of Lewy body in the neurons. But the main difference is this. In Lewy body dementia, the patient presents at the beginning with dementia. So memory loss, kind of maybe paranoid delusion, sometimes often hallucination. Whereas in Parkinson's disease, the patients usually first present with motor symptoms, so slowing of movement and tremor, etc., etc. In Robin Williams' case, apparently the doctors thought he had maybe early Parkinson's because he had a slight tremor. But as it turns out, because of all these Lewy body symptoms and also the dementia that he basically had because his memory was gone, it's likely Lewy body dementia. So that brings us to today's article. So we do not know. So for both Parkinson's disease and Lewy body dementia, we really don't know what the cause is. About 10% of Parkinson's disease has been linked to genetic mutation. But the remaining 90% is what doctors, clinicians call idiopathic. It has no known origin. But we know there are risk factors out there. For example, for Parkinson's disease, I believe farmers are at a higher risk of getting it because of some of the pesticides that they use, such as rotenone and paraquat. For Lewy body disease and for Alzheimer's in general. So for Alzheimer's, Vincent talked about it a little bit. It sounds like the emerging evidence suggesting herpes virus, the herpes family, I don't know, species, class, whichever, herpes viruses might be a risk factor for Alzheimer's dementia. So dementia from Alzheimer's is the number one cause for dementia. The second most prevalent cause is Lewy body dementia. And we, broadly speaking, don't know what some of the environmental or genetic cause of Lewy body dementia is. But this paper that we are talking about today kind of zoomed in on one potential culprit, and that is air pollution. So apparently, there has been ongoing evidence. People are still collecting it, and in science, evidence kind of goes up in a Bayesian kind of way. As you collect more evidence, you're more certain about your conclusion. So there's ongoing evidence that a lot of dementia, including Alzheimer's dementia from Alzheimer's disease, seem to correlate with air pollution. And in this paper, they looked at the other type of dementia, which is Lewy body dementia. And what they did was, so we're jumping into enough pre-talking. We're going into the paper now. So what they did, the first experiment they did is an epidemiological study. They took in US, so we grumble about US being very insurance-based, no public health infrastructure, but there is Medicare and also Medicaid. So after you retire, so correct me if I'm wrong, but I believe after you retire, your employer no longer sponsor your healthcare, and you go onto this thing that is government sponsored called Medicare. And because it's government sponsored, there's a centralized database. So these researchers they collected and analyzed the previous, or the Medicare record from, I think, like 2003 to 2014, something thereabouts, that encompasses about 56 million individuals in the US, with across the entire US, mainland, I guess what you guys call, what is it, contiguous continental US? I don't know what you guys call it. Basically, excluding Hawaii and Alaska.
Continental US.
Yeah.
They looked at all the US, and they know, based on the Medicare record, whether if the patient falls into these category, whether the patient had Parkinson's disease. And if the patient has Parkinson's disease, they divide Parkinson's disease into two different subgroups. First subgroup is Parkinson's disease without dementia. And the second group is Parkinson's disease with dementia. And the fourth category is this thing called dementia with Lewy body. Okay, this is a bit confusing, and I would only say this once for completeness, but this is super confusing. I apologize. This is how clinicians do their stuff. Well, we've been talking about this thing called Lewy body dementia. Lewy body dementia is an umbrella term that encompasses two different things: dementia with Lewy body and Parkinson's disease with dementia. So those two depends on whether you present with cognitive problems first or motor problems first. But for simplicity, we will just lump them both together as Lewy body disease, I think, for majority of this paper. So when I say, Lewy body dementia, even I've been getting it wrong. So when I say Lewy body dementia, just think of it as having dementia. But again, so that kind of Lewy body dementia kind of separates it from the other thing that we're going to contrast it with, which is Parkinson's disease without dementia. So Parkinson's disease without dementia is mainly motor symptoms, whereas Lewy body dementia is mainly cognitive dementia symptoms. One very quick stat, which is patients with Parkinson's disease, apparently around 80% of them eventually develop dementia symptoms as well. So this is something to bear in mind with, is that a lot of Parkinson's disease patient or PD patients, they might just not yet have dementia. But for the purpose of this paper, it's PD without dementia versus Lewy body dementia. So having all these kind of diagnoses from all the patients, so you're either healthy or you have Parkinson's disease. If you have Parkinson's disease, maybe you have Parkinson's disease without dementia, or alternatively, you have Lewy body dementia, even though it's complicated, but we'll ignore it. So they have that. They also have your zip code, so they know where you live in the US. And they also have things like your socioeconomic status. They know what your race is and your age. There are all these kind of potential confounds that can correlate out. But the zip code is the most important, because the second data set that they got was your air quality based on, apparently there are like 2,000 plus air quality monitoring system in the US maintained by the Environmental Protection Agency, the EPA, if that still exists.
Who knows if it's going to exist? It's being destroyed. It's just scary.
I actually don't know how much Doge cut it, or like whether, I mean, there's a huge shutdown now, so there's no data collection ongoing, I'm sure. But but there's been during the same time period, the same tenure, the EPA has collected all these air quality from thousands of stations around the US. So now you can have basically a plot of every single county in the US, how bad the air quality is. And by air quality for this particular paper, they mean this technical term called fine particulate matter, I believe is what they called. Fine particulate matter called, and they shorten that to PM 2.5, particulate matter 2.5. And the 2.5 stands for, this is basically an air pollutant that is smaller than 2.5 micrometer in diameter. So just for scale, a human hair is roughly 100 micron wide. So this is like maybe 1/40th of a human hair. So it's very, very small. But it exists and it's floating all around us. So as someone who lives in New York, in Manhattan, right next to a power station, and also 1 block away from FDR, from a giant highway, I read this paper with some dread. This is all around us. And but depending on whether your city or your house is kind of out in the woods or in the middle of the urban area or next to kind of heavy pollution, you would have varying level of this PM2.5 pollutant. And in the map that they showed, you can see in the East Coast is particularly bad. So like around, I'm not good with US geography, but like around Pennsylvania, New Jersey, New York, it's super bad. And then as you go south,
Michigan,
as you go west,
but Maine looks good. Maine, New Hampshire, Vermont looks good.
A lot of trees.
I love where up north where the maple leaves are. Looks great. The Rust Belt, by the name you can imagine, is super bad. Like in the last during last 20 years, and also California.
But the worst place is LA.
I'm really surprised.
It's black. It's like black. You could not, like, it's saturated.
California is super bad. I'm a little bit surprised. What's really interesting, like well, it's also the geography of the place. Like the mountain is
like it's in a bowl.
Yeah.
I mean, we know that the mountains change how the weather patterns are down there. So I wouldn't. I think it's kind of a sink.
But I guess LA is also famous for its smog from like traffic and stuff like that. But this also accounts for several things that can emit this PM2.5 air pollutant are traffic, heavy industry, but also things like wildfires, which we've been having in New York and also LA a lot as well. I remember, like, was it last year or 2 years ago, the wildfire from Montreal, I think, like all the maple leaves, came down, and then even inside the hospital, it smelled like smoke inside the hospital. So that can't be good. So episodes like this can really drive up the PM2.5 air pollution. So you have all these. You have a map of all the counties of how bad things are, and I will quickly point out, it's weird. Maybe you guys, both of you American, can tell me why the counties like on the East Coast are all nice. They're like very, very tiny, and as you go to the west, they just get kind of bigger and bigger. It's almost like as people migrate to the west,
because there's fewer people.
I thought they just said, oh, whatever, we'll just have a big county. We can't be bothered to.
Well, maybe it has to do with like
population size.
There are fewer people, and then maybe at the time there was more farming out in certain places, or ranches, and so blah blah blah. This is all speculation.
Anyway, it's just a huge, like very obvious pattern. But anyway, so after with this zip code geography-based air pollutant pattern, next to it they plotted the incidence rate or the prevalence of Parkinson's disease, Parkinson's without dementia, and also the Lewy body dementia. And based on these data set, they can next try to, as Vincent said, correlate one with the other. So this is an observational study. You have data that you have collected in the past that you can correlate. And what the researchers found is that if you look at just Parkinson's disease, which clumps together Parkinson's disease without dementia and also Parkinson's disease with dementia, there's actually a significant correlation between air pollution and your risk of dementia. Your risk of getting the disease, which is Parkinson's disease, So Parkinson's disease by itself is correlated with air pollution, and they express it as a hazard ratio. So like per increase of 5 microgram of air pollution, you have a 10% increased risk of getting Parkinson's, for example. However, when they stratified this Parkinson's disease into Parkinson's disease without dementia versus Parkinson's disease with dementia, which is similar to Lewy body dementia, they find something quite striking, which is when you look at the risk of getting Parkinson's disease without dementia, it is actually smaller. Whereas if you look at the risk as a function of air pollution, whereas the risk of getting Parkinson's disease with dementia as a function of air pollution, it shot up. So it suggests really that the most of the effect of air pollution is increasing the risk of driving Parkinson's disease with dementia. However, if you look at even Parkinson's disease without dementia, there is a significant effect of air pollution. So air pollution still significantly correlate with Parkinson's disease even without dementia, but it correlates much more strongly with Parkinson's disease with dementia and also with Lewy body dementia. So this kind of caused the researchers to conclude that we probably should look. So first of all, they conclude that there is a correlation, at least in the US, between air pollution, specifically this PM2.5, these small particulates, with Lewy body dementia. So most study would stop here and wrap up and publish a paper. And as Vincent pointed out, this would be observational study, which is pure correlation. But it doesn't tell you anything about causation. So the researchers for this paper, they didn't stop there. They started there. So this is figure 1. So,
by the way, Tim, I was just thinking that it would be nice to look at if there's a correlation between pollution and something else. But in fact, the PD without dementia is the control in a way, because that's low. The correlation with pollution is low. And then when you just look at PD with dementia, then it goes way up, the correlation. The hazard ratio, right?
That's true. But the control kind of, so they have a different disease to study, yeah. So they have a comparison. I mean, the control would be people who didn't even get PD, like the healthy control, perhaps. But it will be on a gradient, so they're going to correlate.
Well, that's in the data. There are people in the study who didn't get PD, so that's where the correlation comes from. I'm just curious if air pollution causes everything,
But I was going to say it's probably it's correlating with a lot of things.
But that doesn't rule out there is a third factor that everything correlates with that actually explains everything.
Right.
So it could be something that correlates with air pollution that actually is driving everything. So to really establish air pollution itself as the driving risk factor that causes things, ideally the researchers would do something that we can't do because it's unethical, which is grab a bunch of people, separate them into 2 groups, and one group of people you give them this air pollution and the other group you don't, and then you observe them, which you can't do. So they did maybe the next best thing. So which is they did exactly what I said in mice. So the researchers, what they did was they went to some of these kind of air collection devices, like from the EPA, but they also actually went around the world. So around the world, every country has their own air monitoring system. And all these pollutants get trapped in these air filters in these machines. And what you can do is you can grab the air filters out. What the researchers did anyway, they grabbed the air filters out and they trapped out all these PM2.5 particulate matter particles. They cut it up into tiny bits, and then they soaked them in organic solvent. So a combination of methanol and dichloromethane, something like that. So it dissolves all the fine particulate matter, and then they evaporate out all the solvent. So all you're left with is the pollution in particle forms. And then you dissolve your all these pollutant in PBS, which is just a standard
science water.
Very good.
That's what I call it, science water. Although we do also use like molecular grade, like pure,
which is also science water. Science salt water, science seawater. This is science salty water. They dissolve it in science water, and then they actually find mice, wild-type mice. The first experiment, they looked at mice, and they pipetted it into the mouse's nose every 2 days for 10 months. So instantly, the first critique is, what on earth are you guys doing? Why don't you just grab the polluted air and make the mice breathe it? But as it turns out, logistically, it's very hard. You got to go and grab like 10 months long of air supply for these mice to breathe. So it's really difficult. So the authors did kind of the next best thing, which is they pipette it into the mouse's nose, and that would kind of expose the mouse to, they admitted, a much higher dose of the air pollutant than people are normally exposed to. This is 10x higher, and it is once every 2 days. So it's kind of like a pulsatile treatment of high concentrated air pollutant, as opposed to a chronic exposure of lower concentration. So this is one drawback that they themselves point out, but because of logistic reasons, that's the best they can do. Maybe there are some researchers out there now who are leaving a cage of mice by the roadside and come back and come back in 10 months and see what happens. But for this paper, they had to go this way. So this is definitely one of the criticism. But what happens, so that criticism aside, what happens if you kind of nasally irrigate the mouse with these air pollutants for 10 months? What they did was this. They, after 10 months, they euthanize the mice, and they look at the brain kind of gross morphology, kind of structurally, what does it look like? So they have two groups. They have the mice who got the control treatment, which is just PBS, the science water in the nose. So these mice don't show any kind of weirdness in the brain. The experimental group, which got the PM 2.5 air pollutant in liquid form into the nose for 10 months, they show huge holes in the brain where the ventricles massively expanded. It is actually
that's what I was shocked.
I too was shocked. So in the kind of in the pictures they show, these ventricles like blew up hugely, and the cortex shrank majorly, and like it seems like the amygdala might have completely gone, and also some of the basal ganglia structure, like the GPE.
Yeah. So what do you think about that, Tim?
And also the hippocampus volume also dramatically reduced.
I didn't see that though in the pictures.
We have to trust
I don't know if like
we have to trust them.
I don't trust anybody. I trust no one. But
according to them,
that was shocking.
According to them, the hippocampus volume also dropped. So it is what it is. If it's true, and I mean they collected the data, it is what it is. And they're going to tell us maybe how it correlates with some of the symptoms, some of the molecular symptoms that we're going to go into. But I definitely was a little bit, I'm like surprised, because these are quite big changes in the brain. Like they're just huge holes. There's no two way of saying anything.
Yeah.
So they also, in addition to looking at gross morphology, so they showed this to be, so this is the gross morphology. They actually have a second experiment with mice that have a mutation that knocks out their alpha-synuclein. So this is a quick, I'm going to make a very quick digression to talk about alpha-synuclein. So alpha-synuclein is this protein that you make in your neurons, also in red blood cell apparently, but we'll ignore that. Your neurons make this protein called alpha-synuclein, and normally they exist in the cytoplasm and apparently in the nucleus. So synuclein is based on two words, synapse and nucleus, because alpha-synuclein the protein alpha-synuclein is usually most concentrated, according to people, in the synapses where neurons talk to each other, and also in the nucleus in a soluble form. But when something goes wrong in Parkinson's disease, with or without dementia, and also Lewy body dementia, you see these alpha-synuclein start clumping up into Lewy body. And Lewy body have both alpha-synuclein and also a bunch of like weird lipids all glommed up together. And people, the hypothesis is that the Lewy body, actually the alpha-synuclein in the Lewy body cannot be appropriately degraded by your cell's protein degradation machinery. So you're suffering from indigestion. And which is a similar kind of hypothesis people kind of have with other protein aggregation diseases, such as maybe amyloid beta, which is extracellular, and also like maybe phosphorylated tau, I think from aggregate, although don't quote me on that, I know a lot less about it. So these alpha-synuclein, there's several things that are of interest to us for this particular paper. First of all, is when you have diseases, especially with Parkinson's disease and also Lewy body dementia, and also a different disease called multiple systems atrophy, MSA, which we won't go into it because it's not related to this paper. People have increasing evidence that these alpha-synuclein proteins, they not only do they clump up together, they clump up together in such a way that they causes more normal alpha-synuclein to form extra clumps, and these extra clumps can spread from neuron to neuron, or maybe from mouse to mouse, to themselves propagate these clumps.
Kind of like prions.
What I've just described is a prion disease. So these are malforming, kind of misshaped or malformed aggregated protein that are clumping up together that first of all cause disease, and secondly can transmit.
Right, but they're I was just thinking, I was like, oh my God, so like if you stand next to somebody who has Parkinson's, no, they're talking, Tim, they're talking about, when we say it's like communicable, it's like they are injecting the protein that's been isolated from a mouse into a mouse that didn't have the condition to begin with, right? It's not like one mouse coughed on another one and then So don't worry.
So yeah, exactly. So this is a huge. I guess we should take a PSA now, which is this prion diseases, as far as we can tell, is not transmitted by air or even by touching people. So, for example, the original prion disease characterized by Stan Prusner, which got him a Nobel Prize, was in on mad cow's disease, basically. So that involved feeding cows to cows and also sheep to cows. And in
you're mixing up two things, Tim.
Am I? So there's scabies.
So Prusner worked on scabies, which was just transmitted among sheep.
Okay.
But the mad cow disease epidemic was caused by feeding sheep to cows.
And then apparently cows to cows, maybe. I don't know. It got messy. Yes. But it is caused by the same pathogenic agent, which is this protein called the major prion protein.
Yeah.
And that got transmitted to humans when humans ate diseased beef, basically. And the scary thing is you can cook your burger to as high as, well, maybe not as high as you want, but to a normal, well-done phase, well-done cooked. I don't know what the culinary term is. Make it well done, but you still get the disease. You still get the disease because the protein remain misfolded, and it would make your own protein misfold. So that's one of the, but you can only get it, as far as we can tell, by either eating the disease burger or by having some sort of perhaps kind of blood contact with the protein, with a malformed protein. So I know about this because there was a case maybe a couple of years ago where a French undergrad got mad cow's, quote unquote, mad cow's disease, like CJD, the human form of mad cow's disease, when she was volunteering in a lab that was working on mice that had a mutation that make them have prion disease. And I think she cut herself on a cryostat or something, like when slicing brain, she cut herself. And that turned into CJD, the mad cow's disease, in like 10 years later. It was a complete tragedy. Yeah.
Oh my God.
So we have to be careful when working on these we should be cognizant, at least as neuroscientists. Anyway.
Oh my God.
So one of the characteristic. So we're going to go. So let's go back to alpha-synuclein. So the first protein that shows some of these kind of, first of all, misshaped clumping together property, and second of all, transmissible property, is in the major prion protein that Stan Prusner worked on. The second protein that shows this property, discovered by Stan Prusner, I believe, like maybe 10 years ago, was alpha-synuclein. So what happens in alpha-synuclein is this. First of all, you can extract brain tissue, post-mortem brain tissue from humans that died of Parkinson's disease or Lewy body dementia, and run Western blot on it, and which basically separates protein into different sizes. And then you stain for alpha-synuclein. What you see is, in disease brains, the alpha-synucleins have much higher molecular weight compared to in normal brain, suggesting that they're forming maybe clumps, maybe polymers, oligomers. Secondly, when you subject to these proteins to proteinase K digestion, so proteinase K normally would just go into your protein and start chewing up all the peptide bonds and turn it into kind of peptide monomers. So what you see is, as you expose these proteins to proteinase K, before you run the Western blot, your signal goes down because all the proteins, all your antigenic sites where the Western blot antibody label would recognize, they get eaten up. So you can't label for them anymore. Well, in diseased brains, when you try to do the same thing, proteinase K doesn't work. It doesn't digest these clumps very well. It suggests that these proteins have clumped up in such a way that it makes proteinase K really hard to access them. So they're forming misshaped clumps. And thirdly, another characteristic these things have is, if you get brain tissue from disease patients, or even the CSF, the cerebrospinal fluid, which you get from spinal tap, you can put in a bit of these patient samples with alpha-synuclein monomers. So these are alpha-synuclein that you get by recombinant biochemistry. So you kind of make bacteria grow these alpha-synuclein proteins and then purify them. So then it's just a pipette full of dissolved alpha-synuclein. And if you just add PBS to this pipette or this tube, this tube of alpha-synuclein, and let it sit around, nothing much happens. But if you add some patient samples to this tube of monomeric alpha-synuclein, what happens is they start forming clumps. So the hypothesis is that alpha-synuclein from patient samples, because they themselves are misfolded and aggregated, they act as a seed, and they make the monomers in your tube that are normally dissolved, they start clumping up. And you can detect these clumps by kind of putting in a fluorescent label called THT, which is a thioflavin marker, fluorescent thioflavin marker, which doesn't matter. But these labels are fluorescent when they kind of intercalate, so they wedge themselves into beta sheet structures. So beta sheets are these secondary protein structures that are found usually in protein aggregates that form in kind of this amyloid form that we see with amyloid beta, for example. But when protein clumps up in this misshapen neurodegenerative form, they form these beta sheet, and therefore you can kind of detect them using this THT labeling. So patient samples can make a test tube worth of soluble alpha-synuclein, or quote-unquote normal healthy alpha-synuclein, start forming these clumps, these misshapen clumps. And you can then pipette out some of these misshapen clumps, and they themselves act as seed. So it is transmissible first of all, to a new test tube. So you can make a new test tube, start forming clumps. Not only that, you can get some of these so they call it seeding amplification assay. So this acts a bit like PCR, but with proteins. So you can use these seeds to amplify to get more seeds. And you can get these, and the result is what they call a preformed protein fibrils, PFF. And you can get some of these clumped up fibrils and inject it to a mouse. So we discussed this previously in TWiN 30. You can inject these misshapen fibrils that you get from a test tube into a mouse's gut. And researchers have found that this would cause your gut to have disease. You can stain for alpha-synuclein phosphorylation, which correlates with the disease state. And this disease pathology would go actually up your vagus nerve to your brain. So it would spread from the gut to the brain, which is one route that people hypothesize how Parkinson's disease is acquired, is that you can get it via your gut, which correlates with a lot of kind of early symptoms you get from Parkinson's disease, like constipation and gut problem. And what is more is once you inject these kind of disease misshapen fibrils that you get from a test tube into the gut, and once the disease spread to the brain, you can get the brain from these disease mice, and they would seed a new round of disease. So once again, it's transmissible. So that whole cycle propagate based on misshapen, misfolded, aggregated alpha-synuclein.
And the air pollution causes the aggregation to begin with, right?
Exactly. So if all that is true, I'm actually running out of time. I better speed up. But we're setting the groundwork so we can speed up in a bit for the rest of the paper. But if that is true, then alpha-synuclein is the main kind of thing in the middle that propagates all these disease. It is almost one of the driver of the disease. And if that's true, going back to our mouse, which got exposed to this PM2.5 pollutant, if alpha-synuclein is very important, then in an alpha-synuclein knockout mouse, they should not show any of these symptoms, or at least these symptoms should be much reduced. And that is exactly what they saw. So first of all, surprise, alpha-synuclein is actually not necessary, so you can knock it out. And the hypothesis is that there are other kind of synuclein that can gets upregulated to compensate. So like beta and gamma synuclein gets upregulated. But if you knock out alpha-synuclein, the mice show protection against the, massively enlarged brain ventricle and also shrinking of the cortex in a hippocampus. The second thing so this tells you that alpha-synuclein is very important, is probably kind of the one of the driver of all the pathogen. But the second thing that the researchers looked at is phosphorylated alpha-synuclein, which is one of the markers in disease brain, in these Lewy body, where all the alpha-synuclein clumps up. You see a lot of hyperphosphorylation of alpha-synuclein at this site, serine 129. So lo and behold, when you expose mice to PM2.5 to the air pollution, you see much increase of this phosphorylation. Whereas you don't see any in the alpha-synuclein knockout because the protein is not even there to begin with. And then, not only that, in these air pollution exposed mice, they have increased phosphorylated tau, which is a marker of Alzheimer's disease, funnily enough. So it also correlates with dementia, and also frontotemporal dementia, which is another type of dementia, also has increased phospho-tau. But interestingly, tau is hyperphosphorylated, similar to Alzheimer's disease. But dissimilar to Alzheimer's disease, there's no increase in beta amyloid. So that is another marker for Alzheimer's disease, is that you get these plaques, which is rich in this protein called amyloid beta, or beta amyloid. I can't remember which one is. It's one of those.
I think they're both
used. But you don't see it in with air pollution. So that tells you it might be slightly different. There's a different form of brain degeneration. And in addition, all the glial cells got activated by air pollution. So there's an increase in microglial activation, and you can tell by the morphology. Normal microglia, quote unquote normal, Vivian might start standing on her soapbox, but quote unquote normal microglia show these a lot of kind of proliferation of like not neurites, but they have a lot of, I guess, processes. They like spread the tentacles everywhere. But when you cause an injury, you can beam like shoot a laser in, or you give it some stress, like for example, air pollution. These tentacles kind of shrink and they switch form into a different activated form. And that's what they see with PM 2.5 exposure. And this is protected in alpha-synuclein knockout. So, and separately, astrocytes also get activated. So whenever there's any brain injury and neurons start dying, so if you get apoptosis of neurons, what you get is astrocytes start proliferating. We call this gliosis. And you see this with PM 2.5 exposure. You get many more astrocytes. And this is, again, protected in alpha-synuclein. Vivian.
I just wanted to say that I think, the figures associated with those findings are in the supplement, I think, right?
Yes.
Yeah, and so the astrocyte staining is pretty remarkable. It's huge.
Like huge upregulation.
Huge upregulation, yeah. But I think it is, it's difficult about GFAP, and what's difficult about cytoplasmic stains, and if you're not doing confocal, is that it's hard. If you have a cell that has a lot of different arms and branches, it's hard to tell, oh, is that a cell, or is it just like the arm from another cell? And so, you can have the upregulations. It doesn't really matter, I guess, for the takeaway message, but that it could be that it's massively upregulated. It is, and then it's possible also that the morphology and also the number of astrocytes is changing. Okay. But I just also thought I wasn't really convinced the microglia findings didn't make a ton of sense to me. Maybe it's just a problem where they just chose the wrong representative photo of their conditions, but it actually looked like there were fewer in the images they showed. It actually looked like there were fewer microglia.
I think they went with how branching are each microglia as opposed to number of microglia.
But that's just one measure.
Yep, that's right.
And oftentimes when you have if there's a microglial population expansion after in inflammatory conditions, especially if there's an attempt to try to clear things, or if there's an inflammatory phenotype that pops up, then those cells proliferate. So, maybe a future study would be to dig into understanding what's happening with the glial cells, since, they're going to drive a lot of the kind of collateral damage that we see to neurons and neurodegenerative disorders. So, yeah. But I thought that was I thought it was a little weak.
We will go back to the immune angle right at the end when they look at transcriptomics. But I guess I can't speak for the authors because but I don't know.
No, you shouldn't, right? But the point here is,
But that's one potential kink, is that apparently, So something to watch out for is that maybe the microglial numbers are down, or at least they didn't report it. But finally, what the last remaining glial cells they looked at was oligodendrocytes, and apparently the myelination, some of the markers of myelination might also be down. And this might make sense because if the neuron number is down, then there are fewer neurons to myelinate. And in fact, neurons numbers are down. Did they show it? Maybe. I don't know whether they're labeled for TWiN, according to my notes, not for this experiment. But they did. They labeled for double-stranded DNA break. So this is one tunnel using this thing called tunnel assay. I won't go into it, but this labels for when cells undergo apoptosis, which is programmed cell death, the DNA gets fragmented. So you have a way of detecting it, and you see more of it in air pollution exposed mice. And this is protected once again in alpha-synuclein knockout mice. And also there's more oxidative stress, et cetera, et cetera. So a wide host of kind of molecular and morphological changes in mice that are exposed to this air pollution for 10 months. So this and almost all of it is protected by alpha-synuclein knockout. So the next thing the authors looked at is, are there any deficits in behavior? Like what happens when you're missing half the brain? At least the ventricles are drastically enlarged. So some of the things that they tested was nest building, which is, they say it's a cognitive. It definitely demands, we don't know what it demands in a mouse, but it is impaired in mice that are exposed to air pollution for 10 months.
I mean, I think it's generally used as just a kind of like measure of well-being, because they'll also use it in like stroke studies, or we briefly toyed with the idea of using it as a measure of mouse well-being after infecting them with the virus. And I think it involves a lot of things that are kind of subcortical as well,
it involves,
and like hypothalamic
for sure.
and stuff.
Yeah, it involves many, many things. And I think like Rob Fromke in NYU showed that like mice who don't have good oxytocin, who are not very good at parenting, they make very horrible nests. And in our hands, mice who are Parkinson's that show motor symptoms, they also make horrible nests. So it's a lot of things.
The 3xTG Alzheimer's mice also make terrible. So they don't care.
It's an umbrella thing of that there's something wrong with these mice. But
but not very precise.
Not super precise.
That something's wrong.
But these authors kind of zoomed in a little more, where they tested the mice on some memory task. So they have this thing called Y maze recognition. So the mouse have to go into a Y maze. One of the arm is blocked. So the mouse would explore just one of the arm, and then you remove the blocked arm, and you see if mice go and check out the new arms. And normal mice would check out the new arms. If you have memory impairment, maybe you forgot which arm you checked out previously, and you're more kind of indifferent. And that's what they saw with the pollution exposed mice. And another thing the experimenters looked at was this thing called novel object recognition, which is you just basically put in object A in a box, show it to the mice investigate it, and then you take the object away, and then you put in two objects, the old object and a new object. And normal mice would spend more time investigating the new object. These mice, if they show deficit in memory, they would again be more indifferent, and that's what they saw. And seems like there's some protection, but the stats is a little bit inconclusive. So there's not significant increase from the wild type mice. Protection by alpha-synuclein knockout. So it's not super strong. Well, it's one of the criticism that they're going to have.
Do they bring up do they talk? I didn't look at their limitations yet, although I should. But do they talk about the fact that their knockout mice are in a different background than their wild type mice? That's potential. It is. They're on a different. The knockouts are on a different background strain. And I only bring that up because I'm sure you guys have seen this too. That if you choose mice on different background strains, they can have very different behaviors at like rest. Like in home cage behavior is really different. So then if they're starting at different places, you can't compare between strains almost. It's more like you have to say, okay, an animal of this background that doesn't have the genetic mutation, how does it compare to an animal where something has been modified?
Yeah, they I don't think they mentioned it too much in the limitation. They largely said that it didn't seem to have a strong behavioral effect. But that would definitely be something to watch out for. Although I would quickly point out that they also have a different group where it is alpha-synuclein knockout who didn't get the air pollution exposure. And those guys look fairly similar to the wild-type mice who didn't get the air pollution exposure. So that's another control group. Okay.
Tim?
You have to go?
Yeah, I'm going to need to go. Here
I'm sorry.
soon.
Okay. Well,
no, it's okay. We spent a lot of time chatting at the beginning, so
we will continue. You can, when you have to go, Vivian, just leave and wait for it to upload, and then you can leave the room.
Okay. Should I just do that now since we're stopped, or should I say goodbye?
We can go. We could say goodbye, Vivian Morrison from New Orleans. Thank you, Vivian.
Thanks, guys. I'll see you next time. Bye.
Right. So hit leave and then wait. Hopefully she'll wait.
Okay.
Okay, go
That's
ahead.
my
Tim.
bad. I spent a bit too long on the preamble.
I talked a lot. I talked a lot at the beginning too.
But yeah, so these mice definitely show both brain deficit, physiological deficit, and also behavioral deficit. So we know that air pollution. So this answers Vincent's earlier criticism of all these observational studies that you just don't know unless you do the experiment to look for causation. And this is what they did. And it caused both neurological impairment and also behavioral impairment. So what they next did is they went a little bit deeper. So alpha-synuclein is important because when you knocked out alpha-synuclein, you protect against a lot of these pathology. So next, the authors actually transitioned a little bit. So this is a bit of a kind of a inside baseball. But as it turns out, the normal mice don't really suffer from alpha-synuclein pathology a lot, mainly because they only live 2 years as opposed to humans, which live 80 plus years and get a lot of these neurodegeneration diseases. So what they did is they actually moved to a new group of transgenic mice that have an alpha-synuclein knocked in. And the alpha-synuclein that they knocked in is from humans that have a mutation that gives them early onset Parkinson's disease. So this mutation, so they only kind of knocked in this alpha-synuclein gene. And what they found is that normally, if you just knock in this gene in normal mice, after 1 year, they start developing pathology. So so that this tells them that these mice are more susceptible to kind of alpha-synucleinopathies, alpha-synuclein-related disease. So what they did is they exposed these mice to air pollution and they re-asked the question, what does it look like in the brain? And not only that, they actually exposed, they spread out they kind of upgraded the experiments a little bit, and they collected air samples from 3 different places around the world. So first one, they collected it from China. So I just expect that to be massively polluted, it being China. The second, they collected in Georgia in the US. And apparently this is a kind of rather leafy residential area, but it does have some light traffic and it's close to an airport. And the place they collected from China apparently doesn't have any heavy industry, but has a lot of traffic nearby. And the third place they collected is from Europe, from Prague in Czechia. And apparently this particular PM20, so all these are PM2.5 samples that they collected. In the last one that they collect from Prague, you can just buy from Sigma, which is a chemicals company. I did not know that they sell them. It's weird that you could just buy air pollution. So they collected it around the world, and the goal is to address the question, is this air pollution link only a US phenomenon? Because in the figure 1 that we talked about, it was US Medicare data. So they exposed these alpha-synuclein transgenic mice that are more prone to the disease to either PBS, which is the control science water that doesn't cause much disease. So for these mice, because these mice are prone to getting the disease, they only expose them for 2 months as opposed to 10 months before. So science water control vehicle didn't cause any minimal pathology after 2 months. But in the remaining group, whether the PM2.5 is from China or from US or from Czechia, Czech Republic in Europe, they caused a very similar pattern of pathology, which in their case, they labeled for this hyperphosphorylation of alpha-synuclein. And this is especially prominent in the cortex and also in the hippocampus and also in the amygdala, a bit in the midbrain. They say it's not in the midbrain, but you can see a little bit of it in the midbrain. Also in the brainstem. And also the olfactory bulb. So one interesting question is, so I mentioned a bit earlier that there is actually a hypothesis that you get Parkinson's disease through your gut by things that you ingest. So for example, pesticide, maybe farmers get it either by working, they will get it through the hands perhaps, but maybe they also unconsciously eat it because of exposure. But a lot of, so there's some evidence, I wouldn't say a lot of, but there's increasing evidence that maybe Parkinson's can be transmitted first through the gut and then spread up to the brain. But there's a separate viewpoint that suggests these Lewy body diseases can be acquired through the nose, because one of the early symptoms is actually you lose your sense of smell, just like in COVID. So the early symptoms of having these Lewy body related disease, which includes Parkinson's and Lewy body dementia, is constipation, loss of smell, and sleep disorder in which you act out your dream, called REM sleep disorder. And losing the sense of smell suggests that maybe you can acquire it. Maybe you see pathology first in the cortex, especially the olfactory bulb, and maybe you somehow breathe it in, which would square with the air pollution theme that we talk about in this paper. So you see also in these mice, there's quite a lot of alpha-synuclein pathology in the olfactory bulb and also in the cortex, which suggests maybe they are more a nasal cortical cluster than a gut cluster, perhaps. Although these researchers also looked at, just to instantly refute my own point, they also looked at alpha-synuclein pathology in the gut and in the lungs, and they saw it in both places. So maybe since they are dripping it onto the mouse's nose, some of it might get into the lungs and some of it would have been swallowed. So maybe that is causing pathology there too.
Yep.
Okay. So now we know that it doesn't matter where in the world these air pollution cause similar disease in the brain. Next, the researchers looked at what we talked about a bit earlier, which is this pathological seeding of alpha-synuclein kind of fibrils, of kind of protein aggregate in a test tube. So what they did was this: they had mice that are, so these are these transgenic mice that either had no exposure to the PM2.5 pollution, so this would be the control group, versus 5 months worth of exposure to PM2.5 air pollution. After that, they extract the brain, and then they homogenize the brain and they extract the insoluble fraction. So this is the fraction where all the clumpy proteins are going to sit. And then they pipette out a fixed concentration and they put it in a test tube of monomers of alpha-synuclein. So the monomers of alpha-synuclein is going to act as kind of like, in PCR would be like the ATCG. They act as the kind of the substrate of this seeding of these clumpy fibrils. And they're going to detect how much fibrils are formed using several assays. The first assay is this THT fluorescence assay that detects clumpy beta sheets. So what they found is in the control group, which is alpha-synuclein transgenic mice, they actually show a little bit of symptoms. So I talked about a bit earlier. These mice are prone to developing a little bit of alpha-synuclein disease, mainly manifesting in like a more motor phenotype, like the dopamine cells are lost and they see like hyperphosphorylated alpha-synuclein. These guys, if you incubate the brain homogenate sample with these alpha-synuclein monomers for 7 days in a test tube, they start forming these clumps, and you can detect it as an increase in this THT fluorescence. That's fair enough. But what is interesting is that in the PM2.5 exposed mice, so these are the same transgenic mice, but you make them breathe, you don't breathe it, you make them exposed to this air pollution particles for 5 months, and then you extract the brain and do everything else the same. You put them in the seeding assay. You get maybe close to twice as much protein clumps, twice as much of these preformed fibrils. So the fluorescence is almost twice as bright. So this is first assay. The second assay is that they got these preformed fibrils that have been clumped up, and they ask how resistant are these clumps to proteinase K digestion. And what they found is that the clumps from the mice who are exposed to PM2.5 are much more resistant to proteinase K. So that tells them that these clumps both have more beta sheets and also are in a maybe different conformation that make them more resistant to proteinase K. And they did also a separate assay called circular dichroism spectra assay, which uses light that tells you a little bit about the structure. And they found that these air pollution gives you a different structured alpha-synuclein fibrils. We won't go into that because that is the extent of our knowledge.
So these experiments, I thought they were done with recombinant synuclein.
Correct. Yep.
Right. So then they add the pollutants to that, and in vitro they're misfolding, right?
That is the next experiment. The first experiment.
That's the next one.
The first experiment, the first experiment had the mouse as the middleman. Okay, so they exposed the mice with the air pollution, and then extracted the mouse brain, and then put it in the test tube, and they formed these clumps. So the next question is what Vincent got to. The researchers asked, is the mouse necessary? Because you can imagine, we talked a bit earlier about all the glial cells that get activated. You can imagine these pollutants can activate the glial cells. The glial cells maybe secrete something that make all these alpha-synuclein clump up, and then the neurons can't digest the alpha-synuclein clump. The alternative hypothesis is that the pollutants themselves can directly somehow modulate the clumping of these alpha-synuclein monomers even outside of the cell, kind of in an in vitro setting. So that's exactly what these researchers did, is that they ran another set of experiments where they just had recombinant alpha-synuclein monomers, soluble alpha-synuclein monomers, and they just pipetted in some of these fine particulate matters, all these pollutant, no cells. And then they shook it for like 7 days, I think. So the same experiment. And they asked, in the control group, they pipetted it in PBS, science water. And they asked, first of all, do I get increased TNT fluorescence? Do I get more beta sheets? And second of all, is it also more resistant to proteinase K digestion? And the answer is yes in both cases. So what this tells the researchers is that the cells are not necessary, which is very, very weird. So that raises the possibility is that these pollutants might themselves act as a catalyst to...
Do you think the pollutants are actually getting into the neurons?
So that's the next question, is that we do not know. Is how do Like either the pollutants get into the neurons and start causing havoc, or the alpha-synuclein have to get out of the neurons, clump up, and then get back in the neurons to cause havoc.
Because there is a blood-brain barrier, and these are pretty big particles, right? That we're talking
so
about
they
here.
would have to make it past the blood-brain barrier. I do not know if they can squeeze in the blood-brain barrier. That I don't know if the researchers address in this particular.
But they do an experiment with cultured neurons, primary neuronal cultures, and then adding the pollutant doesn't do anything.
Exactly. So Vincent got it completely correct. The next thing they did is they looked at culture neurons and they asked the question, can we just add the pollutants? And it didn't work. So strangely, after 10, so you can sprinkle in some pollutants for 10 days. So in mice, they did it for 10 months. So it's possible you just have to wait 10 months. But at least in 10 days, it did not work. However, if you sprinkle in the fibrils, so these would be the test tube where you put in some of these pollutants. So, okay, let's go back. The researchers now have these test tubes where they sprinkle in the pollutants and waited 10 days. And the result is that the pollutants kind of, quote unquote, catalyze these fibrils. These fibrils have the properties of having more beta sheets and are more resistant to proteinase K digestion. The researchers then got some of these fibrils and sprinkled it onto a neuronal cell culture. And the control group is they sprinkled in either PBS, so basically no fibrils, or fibrils from just alpha-synuclein monomers without any of the air pollution, any of the PM 2.5. So even alpha-synuclein by itself would form clumps given long enough time. So if true, your cell is fighting a battle constantly to prevent this from happening. So they also sprinkled in these two types of fibrils. And the purpose is to show that the fibrils formed after exposure to the pollution is a different strain of fibrils, which they call the PM fibrils. And what they found is that the PM fibrils from the particulate matter caused stronger hyperphosphorylation of alpha-synuclein, which is a disease-correlated phenotype, and more downregulation of a neuronal marker, suggesting there's more neurotoxicity. So it tells you that this suggests that these preformed fibrils are important, and they might be different from your usual run-of-the-mill Parkinson's disease fibrils, like fibrils that are not exposed to any of these air pollution. So we are running long, so I'm going to very quickly summarize the rest of the study. I do apologize. The paper is, I think I uploaded the PMC PubMed Central paper, so the draft of the paper is open access. The gist of the story is the researchers then injected these PM fibrils, so the fibrils that are exposed to just the air pollution, into a live mouse. So they're moving back from in vitro to in vivo. And they once again saw all this hyperphosphorylated alpha-synuclein, and also saw some cognitive deficit in these mice. So it tells you that it is affecting the brain as well. And let's see. Sorry, 1 second. Oh yeah, and they also, another thing, the next thing that they did, next experiment they did is they actually redid a lot of the in vitro experiment, but with patient samples. So now they got the brain homogenate from patient. Actually, no, take it back. They actually got CSF, cerebrospinal fluid, from patients. And it is actually recently, one of the recent breakthrough in all these alpha-synucleinopathy, disease with alpha-synuclein, that my PI, Dr. Eun Kang, is also involved with, is using CSF, so cerebrospinal fluid, in patients who are still alive with us, to diagnose alpha-synuclein-related disease. Because before, you can't do it. You have to do it post-mortem. But now they can actually get CSF from patients and then do the seeding aggregation assay that we just talked about, and show that. And they have shown that patients who suffer from Parkinson's disease and also MSA and potentially Lewy body dementia lead to faster, stronger aggregation of this seeding aggregation assay with monomeric alpha-synuclein. So this is a brand new diagnosis that can potentially allow us to detect disease early. And this is important because if you can detect disease early, you potentially can try to prevent it earlier. And with neurodegenerative disease, it's all about early prevention because once neuron dies, it's very hard to get them to come back. So the researchers got CSF from patients with Parkinson's disease without dementia. So this is the one that is less related to air pollution versus CSF from Lewy body dementia. This is the one that is probably, according to the first experiment they did, probably more correlated with air pollution. And the question is, do they also replicate what they saw in the previous study where the air pollution worsened all these seeding aggregation assay? And that's exactly what they found, which is the Lewy body dementia patient sample CSF led to stronger aggregation in this seeding assay, more fluorescence with the TNT, suggesting more beta sheets. And they're also more resistant due to protein kinase K digestion, suggesting more clumpy, more resistant clumpiness. So this whole story fits very nicely. So suggesting to us that the Lewy body dementia, so the researchers are arguing for the case that the Lewy body dementia is actually gives you an alpha-synuclein form that is distinct from Parkinson's disease that are without dementia, and that it is more susceptible to air pollution. So that is what they are claiming so far, which I think we would probably discuss a little bit more. We'll probably need further evidence, collaborating evidence from other labs. But very quickly, we'll touch upon the final figure, which is they looked at transcriptomics from patients that has Lewy body dementia versus Parkinson's disease without dementia versus, I believe, Parkinson's disease with dementia. So Lewy body dementia versus Parkinson's disease versus healthy control. And then they looked at all the genes that are different compared to healthy control for all these diseases. And they asked the question, how about if we go back to mice that are exposed to PM 2.5, to this air pollution? So remember, mice is the experiment where we can ethically do a causal experiment that exposes the mice to the air pollution. Do the mice who are exposed to the air pollution, do they have gene changes in the brain, specifically in the anterior cingulate cortex? But for simplicity, we'll just say brain. The brain gene changes, does it look more like the humans that have Lewy body dementia? And that is what they confirm, is that the gene changes for the mice who are exposed to air pollution is more correlated compared to the control group of mice that are exposed. And also they had a different group of mice that are injected with the preformed fibrils that are exposed to the air pollution. That's also highly correlated with the Lewy body disease patients. In contrast, the mice who are injected with just the preformed fibrils, so that is maybe more of a Parkinson's disease without dementia phenotype, that those mice, the gene changes correlate less with the Lewy body dementia gene changes. So that is further corroborating evidence that Lewy body dementia is maybe more correlated with the air pollution and all the gene changes and pathology that's taking place in the brain after the air pollution exposure. And specifically, researchers can look at, of all the genes that change after exposure to air pollution, what family of gene does it kind of cluster into? And they found it is actually a lot of immune regulated genes. So immune system likely plays a factor in all this. So that is pretty much a whole story. Sorry for jumping through a little bit towards the end.
So I'm really interested in what's in these particles chemically. I'm surprised that in 3 different locations throughout the world, they have the same effect, but I would think they're chemically very different. And
they might be different, but they are from roughly. Supposedly they can be from perhaps similar source. A lot of it is from burning of, I'm guessing, hydrocarbon.
So I wonder if, the particles, you inhale the particles and somehow they're metabolized and make something soluble which can get into neurons and cause the synuclein misfolding.
Although maybe non-soluble thing can also get into cells. I don't know.
Sure, it could be endocytosed.
It can be endocytosed. And also the preformed fibrils, remember, those are very insoluble. One of the properties is that they are insoluble. So they probably somehow get endocytosed. And some of the researchers on the paper have claimed to have found receptors that are responsible for endocytosing these misshapen alpha-synuclein into the cells. As to the air pollutants, I think it's a work in progress of how they get into neurons and cells.
Sure.
Similar to other kind of environmental things that might be causing diseases nowadays, like microplastic, how they affect us. We just don't know. And it's an ongoing project to look at.
Could also be that immune cells take up the particulates and they go into they are able to get in the brain and somehow they cause neuronal changes.
And that might explain how come the immune regulated genes are upregulated. And so the two things to keep in mind. One is the air pollution particles themselves can apparently cause this misshaping formation of alpha-synuclein.
Yeah, that's true. The purified protein. Yeah.
But whether there are potentially communications from immune cells to the neurons that can cause alpha-synuclein to be passed back and forth is an open question. So there are recent articles that suggest that microglia might communicate amongst themselves or maybe to neurons using these nanotubes, quantum tunneling nanotubes, or whatever they call it. So it is ongoing active area of research. Yeah.
So it seems unlikely we can do much about air pollution, but as you said, maybe we can diagnose this early enough to do some therapy, right?
We may or may not be we may or may not be able to do much about air pollution because in certain parts of the world they are electrifying their cars, and cars is traffic. Pollution from traffic is potentially one major cause of these PM 2.5 fine particulate matters. So it's a wait and see. But in addition to it, like burning of biomass can increase it, and even home cooking might be one way these things get released. So time will tell whether there's anything anyone can do. It's a question of public health, which is a very thorny subject, as you probably know, Vincent. It gets everyone riled up. So that's one thing I probably want to quickly touch upon, which is this paper gives it kind of tells you one contributing factor to maybe Lewy body dementia, which is air pollution. But the question is Lewy body dementia caused by just one single thing? Just like, is autism caused by just one single thing, like maybe Tylenol? Or more relevant to our podcast with Mauro that we did before, just, your microbiome. Is it just one thing?
Sure.
It's unlikely. For things that are complicated in the world, it's likely caused by multiple different things.
Sure.
And in fact, if you go back to figure 1, where they ask the question, how much increased risk you have when you increase air pollution by a bit, the increased risk is quite small. It's not like everyone who lives in LA that has very high exposure to PM 2.5 have Lewy body dementia, even though I believe Robin Williams probably lived in LA. He's not representative of everyone in LA. So it is. It's probably a bit. We can't draw wide-ranging conclusion based on just one single cause. It's probably a multigenic cause for a lot of these difficult disease. But it doesn't rule out that a public health intervention, which will affect millions of people, that's by definition what public health is, can improve the health of millions of people by raising each person's risk by a couple of percentage points.
Yes.
So these two things, I think, have to keep in mind in terms of balancing, outrageous claim versus still doing good, like public health and a measurable health benefit intervention.
Yes. I'm worried in the U.S. that the EPA is being dismantled and there won't be regulation of air pollution as we see it. Yeah. It makes a difference for sure.
And there is an actual cost. I mean, on one side of the coin, it's good for the economy to keep burning oil, obviously, because electricity is a higher price thing. But it comes at a different cost later if it causes health issues down the line. So it's one of those things that whoever's in charge have to think about, I guess.
Also, probably should not be burning coal anymore, and a lot of countries still are.
Yeah, that's right.
Because that emits particles. Even tires on the road makes particles.
Yeah, exactly. Like, why do people have to change their tires every 2 years or whatever? Like, where do the tires go? It's going in my lung, living next to FDR.
That's right.
That's sad, but it is. Well, it's good that we're starting to identify some of these things because that comes with.
Sure, this is lovely. This is a lovely study. I think it's really nice. Combination of observational and experimental approaches is really powerful.
Most study would have stopped at figure 1, so they weren't very much at the extra.
It's lovely. Thank you, Tim.
Thanks, Vincent.
That's TWiN 65. Show notes are at microbe.tv/TWiN. If you have any questions or comments, you can send them to twin@microbe.tv. And if you enjoy these programs, please support us. Microbe.tv/contribute. Tim Chung is at New York University. Thank you, Tim.
Thanks, Vincent. And I'll just shout out to Awakenings, the film by Robin Williams and Robert De Niro, and amongst other people. Go check it out. It's an interesting film if you're interested in neurodegenerative disease. And also, there's the letter from Robin Williams' widow that I think I'll send to Vincent to upload. It's open access, and it's very touching, but also sad, recounting of the end of his life. Yeah.
I'm Vincent Racaniello. You can find me at microbe.tv. Wait, is there anything else I have to thank?
I think that's...
Everybody thank? No, that's it.
Yeah.
You've been listening to This Week in Neuroscience.
Oh,
thanks for joining.
Maybe thanks Vivian again,
yes, Vivian Mars. Vivian Marsden is in New Orleans. Thanks, Vivian. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.